We'll see. So my name is Deb Neher, and I've been at University of Vermont for 21 years. I'm a Midwesterner. I grew up in Kansas, northwest Kansas, where it's very dry, shortgrass prairie. So, and my education is I first did a degree in environmental science back when it was not popular. I was only 3 on a campus. And then I did a master's degree in plant ecology at University of Illinois and a PhD in plant pathology at University of California at Davis. And then I was in North Carolina for 6 years, and I was working with environmental monitoring program. And that's where I got started on my career on biological indicators of soil. So I've been working on that since 1990, which you heard was a time, actually, that's when a lot of things really got started. And then I was in Ohio for 8 years, and then I moved to Vermont. So I've seen a lot of different crops, a lot of different soils. I feel like it takes me 6 years in a new place to go, okay, now I'm kind of getting it. Like, when do you plant this or that, and soils? But it also offers a lot of different perspectives. One thing that came up is this whole idea of gamma grass doesn't work here, right? So I always feel like you want to choose what's the crops that are adapted to your area. That's how we do it best. So that works great in Kansas, but not here. So we have to think what species work really great here and work on it. I feel like when we try to do things that are not adapted to our climate is when we run into problems. Because we're always changing the environment, trying to get them to grow. But we need to work with what does well here already. So. And, there's always a lot of complaints about clay, but I'll tell you there's a lot of rich biodiversity in clay soils. So I've worked with those on the West Coast, the East Coast, and you have them up here. I'm sure there's a lot of differences too in them. So before I get started, how many of you have heard of the SHREC, the Soil Health Extension and Research? Not? Okay, several, but there's a lot of people that haven't. Okay. This is the name of our new Soil Health Center at University of Vermont. We had our formal public launch in April, I believe it was. It was this past spring. And we're getting started. We still have lots of grand plans for the future. But I want to, I'll refer to that throughout my talk. But how we got started, it actually started during the pandemic. There were a group of 11 of us at University of Vermont in College of Agriculture, College of Engineering, and the School of the Environment. We all had interest in soil, and so we put together a white paper that summarized all the kinds of, about soil health, what it is, and what different labs can do, and what, and we had one big piece of it was interviewing farmers in terms of how useful is the data that you're receiving? Does it make sense? Is it helping you? And we quickly learned that there were issues. It's like, I keep thinking I'm doing the better thing, but I don't see these numbers changing. I don't see how they're reflecting to management. Bottom line is we found 2 big things that came out that kind of turned into missions or goals for this new center, and that is to try to introduce more biological measures of soil, because that's been a missing piece traditionally. When NRCS got started, it was called Soil Conservation Service. It started after the Dust Bowl, and it was really focused on erosion. But they haven't really gotten back clear into a full-blown embracing of biology. I worked with them back in the early 1990s, and it was like the first time they were trying to sample anything biologically oriented. So biology, and the other was we need measures that respond to management. Okay? So we need something that if we make changes and we measure that, we can detect that. So I'll be presenting some of kind of what we know at this time. We were able to publish the results of this white paper in a journal called Phytobiomes, and we actually got the editor's pick for this particular journal. They really liked it. And our focus really is small and medium-sized farms. So much of our policy And recommendations are based on really large scale that we don't have here in Vermont. And I grew up on what's called a small family farm in Kansas, but we still farmed 2,000 acres. That was small. But half of it was not in production because it was a fallow every summer. Now that I'm a soil ecologist, I realize that wasn't such a good move to go fallow. You need plants in there. And the whole idea was trying to conserve moisture. Okay, let's get down. So I like to start out with soils are alive. That's why we should care about them. They are alive. They are a living entity. If we're saying the word dirt, that's because we don't really care about it. It's not in the place we want it. It's not doing it. But soil is like saying plant versus weed. You respect it when you think about soil. They're really a component of soil. They affect the appearance. They're very intricately involved in the biochemical and physical processes. And so just to capture some of that, here's just a schematic, thinking about we have aggregate structure, microaggregates, macroaggregates. These are comprised of the mineral particles of soil, but there's a lot of biology there. We don't get good aggregation unless we have that biology. If I had to pick one measurement to do, if that's all we could afford to do, I would say do wet aggregate stability. Because it's getting both a physical structure and some of the functions that come with that. You'll learn more about that from Joshua, but also it takes the biology. It takes not only bacteria and fungi, but, you get bacteria have glues, fungi have like hyphal threads, and then you get plant roots in there, and that's what's the network that holds those particles together. On the right is actually a slice of soil from an old field. So it was in annual crops, and then it shifted. They just let it go, and it's grassland at that stage. The blue is just a resin we put in there. But one thing you notice is how porous soil is. All of the blue areas are voids or pores. And some of these larger pores is where you see the mites and the microarthropods moving around. But I'll just say these are the mineral particles that are kind of in this salmon color. The dark is all organic matter that's around it, and you can even see a root right down here based on it. So, and that isn't a core that we took that we did not disturb it. We just took a slice and looked at it under the microscope. So, biology is part of the soil. That soil is alive. They've said the greatest biodiversity on Earth is in the soil. It's also one of the least understood. It's one of what we describe as the last biotic frontier. We only know 10% of the species that live in soil. We know more about outer space and the depths of the ocean than we do our own soil. Just to give you an idea, it's very biodiverse, very abundant. Here's a teaspoon, which is, they say 1 gram, but that's way more than 1 gram. 1 gram of soil is about like a little pea, okay, of soil. But in 1 gram of soil, just a rough estimate, you get 1 billion bacterial cells, 200 meters of fungal hyphae. So let's see, I was trying to think, maybe like a 1/4 mile of fungal hyphae, and then several thousand of different taxa. I just enlarged this text over here so you could read it better. They say 1 teaspoon of soil contains more organisms than there are people on Earth. So that's just always mind-blowing to me. Another way to look at soil life is to think about this pyramid. If you've had an ecology class and they talk about the energy pyramid, and you have the base of a food web and you work your way up, it always is like a pyramid shape. The smallest organisms are the most abundant, and the larger organisms are less abundant, but they have bigger biomass. But we can see bacteria, about what is that, about, we got millions, billion, trillions. This is per square meter, so square yard, about 8 inches deep. So it's working at the plow layer. And noticeably missing here are fungi. I don't know why they didn't put those in there, which is weird. Protozoa, we're talking about 10 billion. Nematodes at about 5 million. Mites, springtails, we got the microarthropods here in this group, and so on. But each of these would be kind of like a trophic group in there. So just trying to impress on you the incredible abundance and the incredible biodiversity. So I like to think about, I'm really into music. I grew up playing flute and the piano and stuff, so I like the music analogy. I really think of soil communities really working like a symphony. We don't want to think about individual species. We need to think about ensembles of species, because it takes that whole ensemble to get the ecosystem function because of interactions and so forth. So here's how the analogy works, I think. And we know if we go to see a music thing, most of these species are inactive at any one time. They're not all doing their thing all the time. Some do it in different times, do it in different space, different moistures, So they may all be doing a function, but they do it at different times and places. And that there's cues that come on, like in this case, the conductor, but in the environment, it might be rain, heat, temperature, cues that say when it is time to play. So they wake up different groups of organisms at different times, and it really takes multiple species to actually play the same function. So, okay, now this is blue gamma grass experiment that we got brought up about doesn't grow in Vermont, but I like this study that was done in eastern Colorado to just illustrate the importance of a complex food web. So in this system, what we have over on the right-hand side is basically biomass of shoots and biomass of roots. So we're thinking about plant productivity here. They just use those units so that they could standardize it. So if we have a plant alone, we can see the results. Here as we add bacteria, we see a little bit of increase in the shoot. If we add the predator of bacteria, a bacterial feeding nematode, we see even greater productivity of the shoot. We add fungi, and then we can add the predator of fungi, in this case fungal feeding nematodes, or we can put everybody in there. Notice I bolded the highest numbers in each of those categories, but notice the more complex food web, you've got greater productivity in this grass. The greatest on the root biomass, that fungi were important. And I mean, this is generalizing fungi. There's a lot of different kinds of fungi. But there would be some mycorrhizal fungi in there, but also probably some saprophytic fungi. If I use the word saprophyte, do you know what I mean? Okay, no, yes, okay. Saprophyte, so when we think of plants are the base of the food web, both living and dead, okay? We need that living plant, which is providing the food to the food web. But then as that dies and you have the residue, then you got to have something that breaks it down and decomposes and recycles the nutrients. So saprophytes are involved in that. They're the ones that are breaking down that tissue. They're working on dead material. It could be animals, it could be plants, okay? Sometimes we call them detritophores because they're feeding on detritus. So I'll probably use that word later. It just is part of my vocabulary. By the way, if there's a word I use and that one blows you away, just raise your hand and I'll be happy to define it. Okay, and just another illustration that really, well, let me back up here. You've probably seen this if you like to fish or if you like the forest, that you see different stages of ecological succession. So, I mean, if you have a clear-cut forest, it's going to be that, but if you let it go, you can have the mature old growth. If you're fishing, you have those that have different oxygen level or the eutrophic. You can get the catfish or the, carp or the trout. I think we all prefer the trout over that. But the same concept can apply in soil as well in the communities. And it goes with, what kind of crop we're using and how long it stays in that. And the concept is that, a schematic of here we have the plant, here you notice living, but also the detritus. And this would be kind of the food web in more of an annual crop. But if we start to shift to more perennial crops, so if we're going into the hay rotation, or if we're doing alfalfa, we start to support more complexity. You have a larger root system, but you also are longer in time, so you have a chance for that food web to establish. If we go even into longer term blessed, this is where I would say we see the late successional. And it ends up native grasslands in the center of the country until about 15 years ago were known to have the highest soil biodiversity of any place on Earth, more than the tropics. So grasslands, there's a lot of diversity there, especially in native grasslands. I did a study in the tropics too. We found tropics have it too, it's just not all in the soil. They have a lot up above the soil in the litter and the epiphytes and everything. So we just tend to be soil centric. You don't find it necessarily in their soil because their soils are kind of depleted and pretty thin. Okay, biology is I thought I'd throw in this. Here, actually I'll back up and say kind of my pattern is I've given you a few basic concepts, then I'll talk about the indices that we do in SHREC as well as inspirational. And then at the end, I hope to get to a case study that I did looking at the extended season grazing and how that affected forage and soil quality. What I realized from the pictures we just saw, that study was done 15 years ago, and we know winters are increasingly getting warmer, so it probably needs to get redone. So sampling, that's the first step, because if it takes a good sample to get some good and meaningful results. Biology is notoriously aggregated, so it's very heterogeneous spatially. And this illustrates kind of three different scales. We have heterogeneity on the large landscape scale. If we zero down to a field scale, it might look homogeneous here, but at a field scale, you see a lot more heterogeneity. And if you look at just that spot that might have that designation as a particular soil type, it has even more heterogeneity because you're thinking about all the plants. It might be earthworm burrows, there's the aggregates and so forth. So how do we sample such a thing? It ended up just a random sample doesn't tend to represent this very well. What is recommended is I recommend either of these two types. One is systematic, so that when you're sampling, say across the field, you want to be hitting, if you're trying to get the overall field, you want to hit hot spots and low spots and kind of get a representative would be somewhere in between. So a systematic would be you take several cores at equal distances across that field. It's best if you use a random starting point. You can use the phone book, you can use some kind of app, whatever, but that just helps remove bias. So you don't say, I'd like to start here, this looks really nice. But you just get a random starting point so you're not necessarily starting at the edge, and because edges are different than what's in the middle. I am not a big proponent of W's and X's because you're oversampling points in there. But so I can go into that more. Another approach that I think works well for soil biology is what I call stratified random. So it might be you take your field or your plot and you kind of, in your mind, identify different strata. It could be just spatially, I want to divide it into 4 quadrats, or, and then within, I'm just keeping it simple, 4 quadrats, and then I randomly locate something in each of those quadrats. That works well too. So it just depends on, the situation as to what. So, and the depth that we go depends on what you're really after. Plant roots or the litter layer or the plow layer. Because plant roots can go pretty deep when you get into native systems. I usually find biology goes as deep as the roots. And we tend to stick to the top layer because that's where they're most abundant. Usually like 6 to 8 inches. I mean, picked that just mostly because it's the plow layer. But I would say when you get into grasslands, it would make sense to go deeper. If you're interested in the litter layer, by the way, the communities in the litter are completely different, really, than what you find in the soil. So, but fascinating. And how we store the samples before we process them is important as well. If you're looking at bacteria and fungi, put an ice pack in the cooler. If you're looking at invertebrates like nematodes, mites, columbella, don't put ice. Just put it in the container so it's insulated. Because we keep those at, just a little below room temperature. So we keep it at 15 degrees Celsius. But the important thing is, biology, remember it's alive. You don't want to set the soil out on your dashboard or in the back of the pickup exposed because that heat and temperature will affect it. You don't want to drop them because that kills some of them. If you just plop the soil on the ground. We did a fun experiment with that once. So you have to treat them a bit with care, but I find insulated containers just a cooler, nothing fancy. Works, and put it in the shade or something. The point is you want to kind of keep it at the temperature that you sampled it. Okay, a complaint I hear a lot about biology is these measures and indicators are very expensive. And why are they expensive? Because they're very laborious. I mean, and so this is something we have to think about as a SHREC lab as we're trying to offer things that can be affordable, that we can do in a reasonable amount of time, provide useful information, but can be affordable. The issue is we can't just see the biology just looking at soil. We've got to get the biology out of the soil so we can look at it. So we call that extraction. And it can either be, we often will put it in some kind of water slurry. Could be, sometimes we have to run a series of dilutions. If we're looking at bacteria, we have to do this series of dilutions so we're down to 1 billionth so that we can even see them because there's so many. So we have to dilute it so this is 1 billionth of the concentration of the original one. We sometimes can culture them. That's the traditional way. And so we could look at who is there. But only 10% of soil microbes can be cultured. Most of them cannot. So a lot of the scientific literature is biased based on culturing. And so that's why we know a lot more about the organisms that can be cultured. A lot of it is we're putting them on an artificial media, and oftentimes we give them too much food. And there's a lot of them that are adapted to low nutrients, and they just simply won't grow. Then if we're looking at microarthropods, we go through either kind of sieves and water slurry. This is what we do for nematodes. And we use these meshes that are very fine that the nematodes will be collected on. And we go through a series of these sizes, and then we put this into this tray with some water, and they'll just swim out through that. And then we can collect the water. But then we've got to work with microscopes then to do the ID. There's molecular tools for plant parasites, but there are not molecular tools for the beneficials, not very many. That's where we need to head, and soil ecologists are well aware of it. If we're doing mites or columbella, like microarthropods, This is just our homemade setup called a Berlese funnel. You just need a heat lamp like you'd use for chicks or something. And then we have a mesh on top of that and then just a funnel, and they'll drop into this container below. So they don't like the heat and they don't like it extra dry, so they run away. And once they hit this funnel, they just fall down. And we can keep them alive or you can kill them. It depends on what you're planning to do. Sometimes we do bioassays. So if we are testing for like disease suppression, we might take the soil straight from the field. We'll plant like 30 radish seeds in it, and we can see if there's, if there's disease happening or not. These would be, they're not emerging, so there's something happening that's inhibiting that plant emergence. Okay, just want to show you a few of the faces of these critters so you have a clue of, what they look like. And it's hard to show microbes because you have to put them at about 1,000 times magnification or higher. And not everybody has the equipment. Bacteria are particularly, they're not very descriptive morphologically. You get round ones or you get bars. You get spheres or bars. But there's a huge difference in their biochemistry. And so that's where a lot of the more molecular techniques have allowed us to get into the details, and also the genetics. The genetics, we were able to separate them. But here are a few pictures of some that can grow in culture. We have different phyla here, and this would be like Pseudomonas. And there's a lot of different Pseudomonas species. There's some that are biocontrols, there's some that are diseases, there's some that are saprophytes, and then our Rhizobium, the nitrogen fixers, fit in the same phylum. This phylum is very, very diverse, and there's a lot of changes in the taxonomy coming out. But these are like little nitrogen fixation factories. I'm sure you know about the nodules. And it ends up, this is an anaerobic process. So those nodules keep the oxygen out of it. But inside of that tissue, you can see little pockets of the bacteria doing the conversion from atmospheric nitrogen into ammonium. Firmicutes are an interest, what?
I said, that's so cool.
Yeah. You might need to ask me a question every now and then just to break up the monotony here. I do get tired of hearing myself talk. But okay, then there's one called Firmicutes. Firm is referring to they produce endospores. It's a very survival spore. And, they can live decades in soil if soil organisms are very I'll just say from the pathogenic side, they're very difficult to control because they're masters of survival. They have all these different kinds of survival stages, spores, and so forth. It's kind of like a seed is a protection for a plant. And so sometimes you have to trick them, and get them to germinate or get them to be attracted to a plant, and then you can affect them. You got to interrupt their life cycle somehow. Firmicutes. Okay, so they have these endospores, and I'm sure you know of some of these. These are beneficials. Here's the BT, that they've incorporated into various crops. Bacillus subtilis is one that some companies sell for biological control. But you know what else is bacillus? Anthrax, botulism. Those are also species of bacillus. And those are problems because, when you have to get really high temperatures. That's to try to kill these endospores so it doesn't cause problems later. So my point is it's really important to think about what species and not to just pool them on, say, bacteria or even this phylum, because there's so much, it's just like clumping things together that it's hard to tease out. Then there's this group that are filamentous bacteria. They used to be classified as fungi, and you'd see them in books printed as actinomycetes, and I know some books still print it that way. And that was thinking they were like fungi, but since we've looked at the DNA, we know they're actually far more like bacteria than they are fungi. They're a lot thinner, hyphae, and so forth. So now the name is actinobacteria. But there are some that are pathogenic species, but they're also, they work really well in very dry soils, more alkaline soils. They're very slow growing. They're kind of the thing that when you smell the soil, it smells like earth. You're smelling actinobacteria, you know. Over on the right, I just show you a few of the different groups of fungi. Of course, we have yeasts. That term yeast is kind of a fuzzy one. It just means it's non-reproductive. We're just seeing the kind of the vegetative state that just keeps getting, because if we let this become reproductive, it would fit into one of these other categories. But sometimes you just never see it that way. But yeasts are really important. You know, we use it for fermentation products, you know, baking or beer, uses Saccharomyces, which fits in this category. There's another group. This group used to be put in the protist group, and it's now classified as fungi. The chytrids. These are aquatic. I don't have any good pictures of them up here, but these are the ones that are a little, you're probably more familiar with. Zygomycota includes like the bread mold, which is Rhizopus stolonifer. And the names of these different groups are placed on what kind of reproductive structure they have. And it ends up that the zygotes, this is called the zygospore, and these two different mating types merge together. And this is the spore where the sexual recombination occurs. Ascomycota, there's a variety. Alternaria is often a saprophyte. It's also got pathogens. I've studied this in like early blight disease on like brassicas is caused by a species of Alternaria. Penicillium is another very common soil-borne pathogen. This always reminds me, a lot of the antibiotics we have in medicine were originally from soil. Penicillium, there's a number of them from actinobacteria as well. And why do we get them there? Well, these organisms, that's their defense. They're producing these compounds so they can, you know, for their own defense against other species, you know, to be better competitors or antagonists, and so make their way. Trichoderma fits in this category. That's often sold as a biological control agent. Not something I want in my lab because it's a very heavy sporulator. If I grow it in my lab, it just contaminates everything and I can't get rid of it. And then mushrooms, of course, they belong in this other category called Basidiomycota. Just a little different. So I just wanted to give you a few pictures so you can put some faces with names. The protist category is always an interesting one. When I was in school, it was just kind of the junkyard of biology, microbiology, because they just threw everything in there that didn't fit anywhere else. And as the decades have gone by, they're learning more like, oh, this is really should be a fungus. This should really fit, you know, this is really, you know, fits in another category. But I think we all are familiar with like amoeba and ciliates and so forth, like, you know, euglena, because we see those in school. These are kind of different functions they play. And symbiosis would mean, you know, it could either be mutualism or pathogenesis. Either way, they have to work together. Phototrophy would mean they're fixing carbon. So photosynthesizing, it'd be like your algae. By the way, the cyanobacteria would be fitting bacteria, you know, so often we just call it algae, but it's really a bacteria. And then we have this saprotrophy, which is degrading or breaking down organic matter. There's a really important group right here called the oomycetes. They used to be classified as fungi, but now they're as protists. The reason they're important is they're major pathogens globally. They're if you've heard of water molds, if you have really wet soil, those can be a real problem, and they're globally distributed. But the problem is if we just do an analysis where we're looking at fungal to bacterial biomass, we aren't measuring those. They get skipped because they're not considered a fungus anymore. So it pays to at least, know a little bit about, keep up with the classification. Okay.
So practically.
Yeah, I'll pause here for a second, yeah.
Practically, what happens if you have too many? What's the implications
Oh, they cause root rots. So, so they can cause major lesions on roots, and they also can cause a lot of wilting. So basically, they affect the plumbing of the plant. It's a water issue. So the one way you can tell is if some difference between just drought and this is a drought is so it's the leaves are a little wilted during the day, but after night, by the morning, they kind of recover. If you'll probably have one of those kind of, these others, if it's a disease, they won't recover. They're still wilted. If you see that, it's worth pulling a plant up to see if there might be a root problem. It might be, mycota, but there's also nematodes can be another issue. I published a paper on a nematode survey for this state. It was the first one in 60 years that had been published. So it's been a kind of an out of sight, out of mind kind of issue. So it's something I'm trying to help people be aware of. Actually, it was the root rots, that's what my specialty was during my PhD. Let's just think about the role of the plant. We brought that up, how important it is to have living plants in the system. And we brought up the term rhizosphere. That's the soil right around the root. You can also get things called rhizoplane, rhizosphere, or this is the sphere. The plane would be the surface of the root, or you can look at microbiology with inside the root. Those tend to be pathogens, but not always. So what kind of interactions do we get between, well, first of all, plants, they're feeding the food web. So we see what we call exudates. What are exudates? Sugars, amino acids, which would be nitrogen source. Those are kind of two big categories of chemicals coming out. Why does a plant produce these? Anybody know? To what? Minerals. Let me give you a hint. How does a root grow? Does it push from the top or does it grow from the bottom down? Bottom down. You got it. So, but how can it do it? It needs some lubrication. Okay? Because it's kind of tough to just stick something that's kind of dry and try to get through the particles. So they're producing these so that the roots can grow into the soil. They're not doing it to be benevolent and feed everybody else. That's just kind of a sidekick, okay? So, because roots are growing from here down, you're not pushing them from here. I mean, that's the same way fungi grow too. They grow from the ends, and so the youngest roots are always at the bottom, and the older roots, and the younger roots are where you have the most activity. Where you're getting a lot of new roots in the fine roots. Not so much if the roots are woody and getting, multiple years old. So there's different kinds of relationships, different groups. Of course, we know a lot, we hear a lot about pathogens, partly because that's direct reduction in productivity. And that's where a lot of the research dollars have gone to over the last decades, how to deal with those. But frankly, the majority of the community is beneficial. And that's often overlooked for the people that are studying the plant pathogens. I have a degree in plant pathology, but my ecology makes I get more interested in these guys. You have those that are truly beneficial. They're assisting the plant in taking up nutrients. They're providing some kind of defense for the plant. They can protect the roots. Let's see. They sometimes will impact the hormones of the roots, so they can stimulate root growth. There's those protozoa, some of the amoeba can do that. We've seen that occur in a number of soils here in Vermont. And then we have those that are commensal, that's just, they're there, but there's really no direct interaction. They're kind of free living. They like their independence, I guess. But that's just the term we use, but no direct effect on the plant or the pathogen itself. Any questions on that? Okay, just kind of different roles they play. Okay. Now I'm going to transition more to SHREC. How are we doing? Okay. Biological indicators. I came up with just an overview of the kinds of indicators we could have. We obviously can't measure all of these, but we have those that measure activity. We have some that are really getting at who is there, the organisms. We have a group that's trying to measure the function, the ecosystem function. And two big functions in soil would be nutrient cycling and decomposition. I often approach people and say, photosynthesis is important. We wouldn't have plants without it, but what's the second most important process in the world is decomposition. We got to have some recycling. Otherwise, we'd be living in big piles of who knows what. We need to break that down. And another function, of course, is plant productivity. Now, we can't afford to measure everything. So my recommendation or my goal, my theoretical, goal out there is to try to think about complementary measures so that we're getting something that measures activity, something that's getting at function to see if our soils are working for us as we hope, and also to think more than just microbes. We need to think multi-trophic because we get different answers if we just look at bacteria and fungi than if we get something that eats them, because that can reduce their number, but by getting the layers of predators and consumers, you're getting a much more complex food web, and they all start to keep each other in check. You get your balances. So I put stars by ones that the SHREC lab is measuring. One is we have a measurement of active carbon, so that fits in that category. This is in progress. It isn't launched yet, but we're going to do a fungal to bacterial ratio. And for those of you who know methods, this is what's called a PLFA method. I have a slide about that later. But look, this gets us at the amount of biomass and the relative. And for function, we have a nitrogen, an organic nitrogen form, but we're also looking at some of the organic carbons. Okay, I put this little note down here. Where we're at in science is we now have tools to tell us who is there, but we still have a long way to go to learn what they're doing. So that's where kind of the current state-of-the-art science is people are trying to figure out tools. Now that we're getting tools to figure out who's doing what in the nitrogen cycle or who's doing what in decomposition or whatever. So this just shows kind of some of the most popular indicators that are out there and what people are doing research on in science. You'll notice soil respiration has the most. That's the one often when like NRCS would just started getting into thinking biology, that was what they wanted to do is respiration. And then the next would be looking at more this active carbon, some that's being mineralizable, and I'm just going to call it POC. This is one we do. We're bringing online respiration. And we also have this type of protein which gets at organic nitrogen. So we're trying to work with the literature and what's the recommendations. And these are, thinking about how they relate to cycles. So while I'm thinking about that, over on the table over there, we have some fact sheets for the different indicators that we do. You're welcome to pick those up. If we run out, we can make more copies, or you can always get it on the web page. All of those are linked to the web page. And they tell you how to take a sample, you know, how to submit a sample, but also what does the data mean. And that's something that I feel like we feel like our filling a need is trying to, how do we interpret this? What does it mean? So one on respiration. So just to think about how respiration works, everything that's living in soil is contributing to respiration. Plant roots are contributing. You got bacteria, fungi, some of the microarthropods. So it kind of pools all that together. But remember that pyramid, the most abundant ones are going to be bacteria and then fungi. These others are less abundant within that. But it's just a measure of the carbon dioxide that's coming out of there. So you get it from the organisms, and I just always say if you got plants in there, they're contributing too. So one of the challenges is hard to interpret. It gives you a general idea of how much activity, but you can't tell who's doing it, how much, who it is. CO2 obviously is an ingredient into photosynthesis, so we can control, more of the plant growth above ground. But basically we see this kind of estimate. If you have low microbial activity, you get a little bit of carbon dioxide coming off. If you have a lot of microbial activity, you get more. So this is it's just the concept of here. This is my own opinion based on my own professional experience, but I think what we want to think about is kind of an intermediate level. It's not always that more is better. Generally, I think, I think theoretically we think more is better, but here's why I disagree with that somewhat, is if a plant is stressed, either due to drought, herbicides, or disease, you will see a big burst of respiration. It's kind of a desperation call. It doesn't last super long, but it can temporarily really raise that rate. So, and if we cultivate, we see a temporary burst in respiration. So we just have to be a little cautious, and this is where taking samples through time is really helpful to see kind of where it rests. So it's a little tricky. It's a good start. It's one of the easier things to measure, which is why it's been used. As an ecologist, one of the reasons I struggle with this one personally is that you don't know how many organisms are there. I like populations and thinking about communities, where this is a flow of a gas. Okay, what effects what can affect respiration? This is just some data that we collected in my soil ecology class. I'm not going to talk too much about enzyme activity, so just pay attention here to the respiration piece. And what we had here is we had a no-till pasture that had been in place for 100 years. That was our reference. And it's out here, the highest respiration rate. If we tilled that one, added a plow and mixed it up, the respiration decreased a little bit this way. And then we took some samples from other farms. These are vegetables that we were looking at. And we either just left them tilled or we'd add compost. So plus or minus compost, adding some, and compost adds organic matter plus the microbes. I mean, that's the reason I'm really pro manure spreading, because you're getting more than nutrients. You're getting carbon, organic matter. You're also getting microbes that feed into it. It's more holistic than just applying straight nutrients. And it's going to make a more slow release. Yeah.
But what about, so one of the things I don't understand, like the difference between compost and liquid manure,
Oh.
being the environment that they are, like the characteristics of liquid manure versus the characteristics of compost, and how do they impact the life here? And is the characteristics of liquid manure kind of antithetical to the life in the soil? And you could just tell me.
Well, I'm not as well versed on the liquid manure, familiar with it because I grew up on a hog farm, but and got teased relentlessly about the odor. But one of the concerns that people have and you need to know is how those animals have been treated. Have you been using dewormer? What kinds of chemicals might be, in terms of what might be in it. I mean, that's what people think of, or if there's a pathogen or so forth, but I'm not sure what I'm getting at here. Compost is a very precise process. It's not just a heap of decaying material. When I talk about compost, it means you have a ratio of carbon to nitrogen that you're aiming for. It's somewhere, 20 to 40. And a certain moisture you're aiming for, which is about 60% moisture. That's where they estimate the most activity at for soil moisture. And you got to deal with temperature. So thermophilic is the way to make the appropriate compost. And that's basically, you make the pile. If you got the right recipe, they'll just naturally, the microbes through respiration, as they're breaking it down, create heat. And that heats up the pile. And then we need to reach a certain temperature. I think I always have to think of Celsius to conversion. Don't quote me, but I'm thinking it's like 131. Thank you. I just had to, I have to publish in metric, so I've got to be bilingual. So we get at that, but like if it's a windrow, we need it like 15 days, 5 turns. And if you do an aerated static pile, you can do it, it's shorter amount of time. That's all about getting the oxygen in there. But what does that do? Why do we do thermophilic?
Pathogen reduction.
Yeah, pathogen reduction. And you remember like those firmicutes that I mentioned earlier with endospores? It's getting rid of those kinds of things, right? The guidelines were developed based on Listeria and Salmonella, the human health pathogens, but the same applies pretty well to plant pathogens and weed seeds. We've tested it with giant crabgrass seed, and if you do the thermophilic, it'll kill them too. But okay, so anyway, we had the open ones are with compost and the solid ones are without compost. So you can see it raised the respiration a little bit, generally here. I just threw it up because it kind of linked it in with some management practices. This slide, yes?
I'm wondering about the tillage versus no tillage. In my
Yep.
mind, respiration increases after tillage.
It does temporarily, yes.
So I'm wondering, is this like, this is on average, like over a period of time, respiration decreases? When is this measured?
Yeah, that's a good point. This was after a few weeks, because this class was for a semester, and we brought in these big monoliths of soil, like big Rubbermaids, full of as undisturbed as we could get from the field. And so day 1 of the semester, we did the tillage, kind of mixing it by hand, and it was probably about 6 weeks later that we made this measurement. So it's not right after the tillage, you're right, you'll see it burst up. But with it being intact, we're not disturbing the soil food web. Here we're reducing some of the predators that are at the higher end of that food chain by dealing with the tillage. So that's what I would attribute it to. But it does shift the microbial community. Tillage, you shift it more towards bacterial dominated. No-till, it shifts it more towards fungal. And that's why we want this ratio of fungal to bacteria, because it will pick up differences in tillage. Okay. I probably have to skip a few slides here. I think you guys all know what organic matter is. But the point I want to make is, there's lots of flavors of carbon. You got inorganic carbon and you've got organic carbon. When we're looking at carbon, if we're doing carbon sequestration, we're interested in the carbon that doesn't break down. It's passive. But there's these two parts. We have labile carbon that decomposes very rapidly. It's like your simple sugars. And so it doesn't stay very long. It's ephemeral. It'd be starches, for example. But one of our measures here called POC is aiming for this intermediate. So it's important in carbon mineralization, but it's in between these two. So it's active, but it's not going to Because if you're just looking at this one, you might just see it for, it depends on what your system is, you might just see it for a couple of weeks, where this, by looking at this one, is a season-long time zone, which is more realistic to what we want. So we can see the whole season. Just showing you the method, we use this color thing. It's potassium permanganate, and it starts out really dark. The more carbon of this epoxy you have, the lighter it gets. It reacts with this, and then we can put it in an instrument so we can quantify it. There's some debate now on how to interpret this, because usually we thought about it as mineralizable. I meant to take that off, but I'll just say why I put it up there. And now we're having more research telling us it might actually tell us something a little bit more about the storage. So I don't like to just say it is biologically active, but it's not the most. It's kind of in the middle. But it does correlate with other measures of carbon. We see significant correlations with particulate organic carbon, active carbon, and microbial biomass carbon. I mean, there's some scatter and noise here, but there's generally a positive correlation with other measures of carbon. This is our webpage for SHREC. If you hadn't caught that before, go.uvm.edu/SHREC. Just some measures of different carbon and management practices here. This was work done in Michigan. They have the only long-term agricultural research station that it's funded by National Science Foundation. It's probably been there for 50 years or more. But they have from corn plus cover crop, corn, soy, and cover crop. So you get your cover crops or with or without switchgrass and miscanthus, so you're going annual to perennial. And these are perennial, to perennial. And, the more woody species, poplar, but you also get native grasses over here, early succession, so kind of intermediate diversity, restored prairie. So it's kind of nice to see how each of those kinds of, this is all in one location, see how the carbon is here. And we can see this one is mineralizable carbon. And over here is the epoxy, the one that we're looking at that's a little bit more intermediate. But we can see that some patterns here in terms of, being different in perennial versus annual systems. And the length of time, the lifespan of some of these. So it is responding to management. In fact, we picked it up because a lot of agronomists have kind of, done a lot of work together, and they say epoxy is the one that's most sensitive to management, more so than respiration. Traditionally, we've always looked at organic matter, and we've done it by loss on ignition. We run it at, we take our soil, we burn off all the carbon at 500 degrees Celsius, and then see what's left. We're discovering that this is less responsive to management than the epoxy. But this is what we have in our databases. This is what we have decades of data on. So it's important. We're trying to do some research where we can link the other metrics to this one so that we can convert back and forth, so that we can keep that historical data. So we still run this. The Ag and Environmental Testing Lab, is that fair to say? Still does the LOI, yeah. So we don't want to lose that, but our new methods, we just need to run like a calibration curve so that if we do a new method, we can go back and figure out what it would be in terms of this measurement of organic matter. We don't want to throw out everything just because we have something different. We want to keep those connections. Nitrogen. Traditionally, we send in our soils into a lab and we get nitrate and ammonium, because those are the two, most abundant plant available forms of nitrogen. And we know, too bad we can't just use nitrogen from the atmosphere because it's 78% of our atmosphere, but it's got to get converted. And this is, when we get our soil reports, this is what we typically get, right? And then we get suggestions on how much more we need to add to meet the demands. Like Cheryl was talking about how demanding corn is, for example. But what we're adding is one that's going to tell us about the organic pool. That's where this ACE protein comes into play. What do I mean about organic is that basically when you have proteins, so that could be from some of the manure, or any kind of organic amendments that you're putting on there. I don't mean necessarily organic versus conventional, but anything that was living, and whether it's hay or a cover crop, whatever. There's proteins in there, and they will break down into amino acids because proteins are peptides, links of peptides. Peptides break down and into amino acids. That's what they're made of, chains of those. Well, amino acid, by its name, it basically, they're producing, when that breaks down, ammonium. So it gets there to a plant available nutrient, but it's going to take some time. So I often think of this as kind of our slow release nitrogen, so that when it's there, it doesn't just, it's all here, and if you can't use it, you lose it. But allows it to stay there maybe over the season or even longer until it can break down and become available, which I think is a good thing. But it's giving a missing piece for the nitrogen budget, so we can look at a more holistic view of that. So, well, this is one that we do offer right now. Okay, just to show you a little bit, this is the same set of management practices. We have ACE protein on the y-axis here in the same series I mentioned before. And we can look at the patterns where we have greater ACE protein in the these are polycultures, I forgot to mention that. So multiple species. These are monoculture perennials, and these are annuals. So we can see there's a big difference between annual and perennial with this. And I would think, thinking about those roots that Cheryl showed you, those perennials tend to have the deeper roots, and they're there all year long. Where the annuals come down, but they're going to, when you take the plant out, the roots go away. Yeah.
So I'm used to estimating nitrogen, like that's in the soil.
Yeah.
And I actually I'm realizing I've never heard do we know how well ACE protein is at getting the total picture?
Well, I know work is being done on that now. I can't cite it directly, but there's always been, if you looked at total nitrogen, for example, doesn't quite match up to ammonium and nitrate. You're always missing something. So total minus that always left you with, you didn't know what that was. So this is allowing us to get more at that black box. Okay, so hopefully, yeah, Joshua, you might want to add something. There was a really good article on it too in CSA News. The news, about a year ago, I think. So it's the newsletter put out by the Tri-Societies, the Crop Society, Agronomy, and Soil Science. That's one of the best ones I've seen as far as being approachable, to practitioners. Okay. Those are ones we're doing. This is one we're working on. We've just run into a lot of snags in terms of getting our bacterial or fungal to bacterial biomass off the ground. Much of which we have no control over. So, but we've got our samples ready to go. But basically, it's a technique called to find fungal to bacterial biomass ratios. We use a technique called the phospholipid fatty acids. What the heck is that? This is supposed to be a picture of a membrane of an organism. And, as we hear in human nutrition, we hear about saturated and unsaturated fats. I mean, Membranes composed by, it usually has what we call a lipid bilayer, so 2 layers. And the kind of signature of these fats, which are saturated, unsaturated, vary depending, a different kind of bacteria have different signatures, a couple different groups of fungi, So they have different signatures, and so we can, it takes a fair amount of processing to get your sample ready for it, but these signatures can help us estimate how much biomass of these different groups are there. And these are just results from the Soil Health Institute showing relationships between fungal to bacterial ratios. The claim, and I agree with this, is that you want higher ratios are better. You want more fungi. Although I'm not going to just say fungi in general, because you really need to think who is there. Is it pathogens or is it beneficial? And one of the explanations for why this ratio, why we want more fungi, is an idea of ecological succession. So when we till something, it tends to be pretty much all bacteria dominated. But if we now change to no-till, we're not breaking up all the aggregates, and we're allowing the hyphae of the fungi to establish. So I kind of think bacteria come in first, then fungi. Also with them is usually their predators. But if you had more fungi, it tells you it's a less disturbed soil. So that's the idea. It tells you a little bit about the intensity of disturbance. So, they're pretty noisy here, but there is a slight positive relationship with plant available water, aggregate stability, and soil organic carbon. But with that kind of spread, it's only telling part of the story. And this is back to, it's important to pick complementary measures. We might not want to know activity, but this gives us one more glimpse into who is there. Tillage is a big one, and that's been known for several decades. This is proportion of biomass composed of fungi, so this would be like a higher ratio. This is water content. I'm just going to emphasize this, but the open circles here is conventional till, and the closed circles are no-till. We can see we have a higher, these are highest here, higher fungal to bacterial ratio than the conventional till. This also shows you some difference on the moisture. I think we talked about that earlier, how that vegetation keeps that soil moister. So, and it shows up in this data nicely too. Okay. I'm going to just fly through a few slides on methods, but then I want to get to this case study. Keep us on time. Okay. Well then, okay. I just didn't want to, I don't want people like having their stomachs growling too loudly.
We can't do anything about it right now.
Okay. I just want to show you some that I think have great potential, but we aren't offering them yet, but I would love to see it in the future. And it gets at, so one of them that gets more at function is if we can look at enzymes. Okay, why enzymes? Well, bacteria and fungi don't ingest food like we do. They do not. They have to digest their food outside of themselves and then absorb it into their bodies. So it's got to be liquefied food. And you need the enzymes to help you break down. And different enzymes break down very specific bonds. You have to get down into the biochemistry of it to understand exactly which style. But so there's a lot of different kinds of enzymes. But let's just say in the nitrogen cycle, there's a lot of different bacteria that are needed to to convert nitrogen into these different forms. We've got nitrification. We've got, it comes in 2 stages. The process of nitrification has a phase 1 and a phase 2. We get mineralization, anyway, and then you can get nitrous oxides where you're losing the nitrogen. But I just put this here. Each of these arrows is kind of like a different enzymatic reaction. And, so we can start to piece together, where most of the activity is or, in this cycle. I just put it there to illustrate that it's not just one enzyme. It takes a suite of them. It takes that symphony and different organisms. And it starts to get us at what the decomposition and nutrient cycling, which are functions, key functions in soil. So it's well figured out as, say, if we want to know how much cellulose is being degraded, or we want to know how much lignin is being degraded, and we know in soil, or how much urea is being broken down. Those are things that we can tell, and it gives you a snapshot of what's happening out in the field now, which is, we could get results within a day with this. There are high throughput methods now available. Just a reminder that enzyme substrates are very specific. Sometimes they get called the lock and key, but they've got to be a matching between the particular chemical bond and that enzyme, and if so, then that is a catalyst for the reaction. The high throughput method comes, you buy a substrate that has a little label on it. In this, when it breaks down, it cleaves or takes that label off, and that's 1 light unit. So if you have a lot of activity, you're going to get a lot of these light units going, and we recalculate it through time. And then we can quantify it through this instrument, where it's recording the intensity of that light. And so we can look at, how fast it's breaking this down. And this is just to show you we can do kind of a we can put multiple samples on one of these plates, and the instrument takes these plates. I'm happy to go into more detail later if you want, but I figured we want to look mostly at what management does. This is a Soil Health Institute set of data where they have, 6 measurements. I was thinking, respiration is not an enzyme, nor is soil organic carbon, but these are enzymes. Anything that ends in ACE, A-S-E, that's an enzyme. Little secret. And they've looked at rotation, diversity, crop count, how many different crops, residue retention, organic amendments, cover crops, and decreased tillage to see if they respond and have different enzyme. So if we look at respiration, we're seeing in their case, just respiration is an increase. It looks greatest with the cover crops and kind of more intermediate with decreasing tillage and residue retention. For soil organic carbon, we see organic amendments is the big player. But we can also see in these enzymes some different responses to that too. Cellulose, the breakdown of it with this enzyme, we're able to detect residue retention, organic amendments, and cover crops because there's a lot of cellulose in the tissues. We can't break it down, but the livestock can. Chitin is the material on the outside of fungi. It's part of their cell wall. Also, your fingernails, hooves, stuff like that. And we can see that's, relating to similar. I'm not as well versed on this particular one, and this one's more with phosphorus. But you can use it that. I just wanted to kind of point out that there are it is detecting differences in different management practices. Okay. This is actually what I've spent my career on, is looking at fauna, the nematode communities. I just go through it briefly, but what are the advantages of going a little higher in the trophic or in the food chain? Higher there is like whatever you're doing, it's kind of integrating all the information below it. So you're kind of getting indirectly information about the microbial community as well as the soil particles. They live longer than microbes. Microbes can reproduce within hours. These guys can take months to years. And based on some study I did, they don't predict each other. Soil, I used to be asked by NRCS, well, why can't, biology is too much work. Why can't we just do chemistry? It's related anyway. And I said, okay, I took it on as a challenge. And in this experiment, I found that soil properties and microbial biomass only explain 23% of the variation that the invertebrates do. So they're telling us something different. And they're linked in with decomposition and nutrient cycling. I'm going to just skip a few. Well, why nematodes? We usually think of them as plant parasites and reducing productivity, but the majority are beneficial. They're feeding on fungi, feeding on bacteria. They also excrete ammonium directly. That's a direct product, but also by grazing on the bacteria and fungi. And they can hold it in their bodies too until they die. This is a project we did a year ago. It's kind of a really rough graph. And just think about, it's in 4 quadrants, just think about where these different treatments are relative to each other. I see perennial crops here and annual crops here, and mixtures. So I know those are really different nematode communities. It's really sensitive to season. So it's important to narrow down on a season and be consistent about that. And these guys are all just different nematodes that we looked at. And it is sensitive to different crops. Okay, they're in different locations. Somewhat, since sand and silt loams come out about the same, but they are different than we see in clay. Okay, I'm going to skip that. Let me just go ahead to something else. Thinking about, this is just a management effect. Here's like a picture of a soil food web. This is in a grassland. And I've highlighted the different nematode trophic groups, plant parasites, those that feed on fungi, those that feed on bacteria, those that are predators, and those that are omnivores, feeding on a lot of things. This is an untilled grassland. All you have to do is till it once, and it cuts off all the top predators. You just basically get this. You get a few of these, but it's just pretty much hollowed out, that food web. And so an index that I really like detects kind of what stage of disturbance it is. But a variation on that index can be broken down into different components. The one that relates to complexity of food webs is what we're calling the structural index. So it's really highlighting the predators and the omnivores and weighting those. There's an enrichment index that gets more at the fertility. Those that are involved in the grazing on the bacteria and fungi. We can look at the fungal to bacterial feeding nematode ratio here too, and it follows very similar patterns as to the microbes. But what this allows us to do is we can start to put soils into different quadrants. The enrichment, I just tried to put it here, and just a reminder of what these different food webs look like. You know, this would be, it's probably more like here. Degraded is like those well overgrazed things. And, but this one would be more like this, and the most complex would be over here. But it kind of tells us how complex the food web, more complexity. Also how enriched it is. If you have really high nutrients, you just put your manure on, it's going to be up here. So a little bit hard to see, but here's some Vermont soils that we did a year ago for our Food Systems Research Initiative. Most of these are pastures, haylands. It's all those samples that you helped us collect. And it ends up, there's some seasonal differences for sure, but it seems like most of our Vermont pastures are lying up in this quadrant, so they're not necessarily degraded, but we have a lot of nutrients, which makes a lot of sense because we're putting a lot of manure, on these lands, and makes it, we get some over here. So I'm kind of thinking maybe that's the Vermont signal. That would be what we would expect, is something like this. We're aiming for something that's more over here, but we're doing a lot of management in those systems. One thing that we sampled before and after the floods in 2023, We're curious what flooding did. And here is before the flood. These samples are here, and we can see the structure reduced after the flooding. These are the same fields as here. It came, and so it's definitely disturbing it. You lose oxygen if it stays saturated a while, so it kills a lot of soil organisms. Okay, DNA, I'm going to skip that. Here's the case study. Well, okay, so food webs are complex. We're aiming for complex food webs for healthier soil. We want to use complementary indicators, not redundant ones, to be more cost effective and tell us more information. And I listed some of the metrics that SHREC is offering now. So just quickly on a case study I did, and I don't think many people have picked it up because it's not cited very much. But we did it, I was calculating, it's like 15 years ago. I can't believe it's that long ago. But it's all Vermont pastures. We had a CIG grant. The CIG was Conservation Innovation Program. So we did this in collaboration with Center for Sustainable Agriculture at UVM. And we started with the premise that we're concerned about compaction, nutrient runoff, and it might affect soil and or forage quality. This is a relatively small study. We only had like 5 pairs of farms, partly with CIG grants. It's more for demonstration than it is research. And I kept saying, we got to have replication. We want to publish this as research too. So we squeezed out as much as we could. We were comparing bedded pack, outwintering, and stockpiling were the 3 treatments that we did. And we made a comparison of those next to a pasture that was on the same farm that didn't have this treatment. So we could see plus and minus treatment. They were the same during the grazing season, but the difference happened during the cold season. And we were measuring soil and forage quality in this. We worked in 3 counties, Franklin, Addison, and Orange. We took a baseline, and then we did spring and fall in each of 2 years. And I have to preface that we had a pretty small sample size, so I have to take a little bit of the results with a grain of salt because we need more sample size to have more confidence. But for soil, we measured wet aggregate stability. We looked at organic matter, nutrients, pH, and we did the soil nematode thing for our biology. So here's the 3 methods. This horizontal line is the control without the method, and then if it goes up, it means it increased it compared to the control. If the bar goes down, it means it decreased it, and the arrow here shows when that practice was implemented. So notice bedded pack came pretty late in the season because they had to leave the material out of the barn for over a year. It was sitting there before it got applied to the pasture, whereas the outwintering was right away and stockpiling. So, you see some differences. Bedded pack was showing a little more increase and decrease, but just keep in mind it's only a 0.05 unit. It's very small change, so basically none. And this is the organic matter. When you see it all pretty small and close to here, we weren't able to pick out statistical significance. We did see some increase with the outwintering and stockpiling. I think this is just, we were too late to pick it up. We needed to continue the study longer. We concluded it's fairly slow decomposition. I mean, that time of year when it's that cold, things don't break down very fast. Okay, for the nematode communities, we found all these pastures that we worked with the practices got over more towards where we want to see them with the higher structure. It's promoting higher food web structure than without those practices. And this is the fungal feeding to bacterial feeding ratio. I'll mostly focus on, we couldn't see any differences there, but in the outwintering, we actually saw that increase, another suggestion of a more complex food web. And I'll just put this up for relative feed value. We also did, the whole suite of things that Dairy 1 does. But I thought you might be interested in this. We basically concluded we didn't see much effect, which actually was good news, because we were afraid we didn't pick up compaction. Oh, I forgot to say aggregate stability, no differences. We didn't pick up the compaction. That said, what Cheryl showed you, I think we have a lot warmer temperatures now in the winter than we did at the time we did this 15 years ago. So it makes me a little concerned about direct interpretation, because I think when it gets thawed like that, you do increase your risk.
And it seems like so year to year, right? Because
Yeah.
last winter was a great year for outwintering and bale grazing, and the year before was like a total nightmare. So there's that.
So we saw a little bit of slightly decreased relative feed value for the stockpiling and the bedded pack. You can see here and down. And a little bit on the stockpiling, a little bit less of the degradable protein. So it seemed like, if any difference, it had a little bit of a negative impact on the forage quality, which would affect animal health. So we concluded in this study that the concerns for compaction and erosion were unfounded, at least at that time. And we saw a little bit of decrease in the forage. I just say we promote a little bit of caution just because we don't have that many farms here. And it was just a limitation of the grant amount. And we need longer studies and we need more farms to really confirm this. But we felt like the results were pretty good news, actually. It was better than what we feared as far as impacts. So I believe that's it. I have some contact information and social media, my web page, which is way too long, but I put it there. I have no control over that. If you go to my web page, you'll see a section called Publications, and it says My Bibliography. You click on that, and then you get this list of my publications, and I put PDF links to all of those. So just letting you know, I do have them available. You can also email me, and I can send it to you, too. That's it.
Thank you.